So I'll live each day with wonder, see the universe through a brand new set of eyes, one after the other.
Thank you for coming tonight on this chilly San Francisco night to Wonderfest and Ask a Scientist. I'm very excited about this special thing we're doing tonight. 4 short talks, 4 speakers, and the chance to ask questions about each one of the topics. I'm Juliana Gallen. I run Ask a Scientist. There's Tucker Hyatt right there. He runs Wonderfest. And I think I can just turn it straight over to you, Tucker.
I, too, am very excited to be here. This is the first event at which 4 so-called science envoys will be presenting their own research and great insight into their specific fields. The science envoys are part of a project that was created by Wonderfest, in particular by a young professor from Cornell named Jacob Bien. And Jacob's parents are here tonight, and I'm so glad they are. The Science Envoy program consists of 8 budding PhDs, 4 from Cal, UC Berkeley, and 4 from Stanford, who were recommended by their own thesis advisors who were themselves great science communicators. And that's the idea of the Science Envoy program. Wonderfest now has a Patreon page where we eagerly welcome your donations for the cause being science. Wonderfest's mission is to stimulate curiosity, promote careful reasoning, challenge unexamined beliefs, and encourage lifelong learning. If you can get behind those goals, please consider a modest or even not so modest donation to Wonderfest. All right, our first speaker this evening comes from Stanford. She's almost Dr. Lauren Popov. She loves everything infectious. Her talk is entitled Viruses, Ancestors, or Aliens. Lauren, take it away.
Thank you, Tucker, for that introduction, and thanks for the opportunity to speak here. Can everybody hear me? Handheld? Louder? Hello? Yeah, we're good? Really close. I'm going to spit all over this microphone. Like Tucker said, I'm the virus component of this talk, and actually I'm working on Staph aureus, which is a bacteria that causes a lot of skin infections, but my first love in microbiology was viruses. And I thought this would be a really cool opportunity to engage you about the world of virology and cover some of the big ideas in viruses together. So what I was hoping we could do is talk about some big picture questions, like what is a virus? Are viruses actually alive, and where did they come from? So these are pretty large in scope questions, as you can see, and we'll try and get some answers tonight, but I definitely will leave you with these 3 big ideas. Viruses frustratingly sort of defy classification in a lot of ways, and they're an incredibly diverse part of the biosphere. And the origins of viruses remain mysterious, and we can really sort of at best speculate about them, but it's fun to speculate. All right, so I guess we'll start with seemingly the easiest question first, which is what is a virus? I know we've got a lot of biomedically inclined and maybe some environmental scientists here as well. So I'd like to actually ask you guys if you have any suggestions or your own definitions of what a virus is or what comes to mind when you think of viruses. Could somebody shout out some viral traits? Parasite, yes, that's a great idea. I think that really gets at sort of the key concept of a virus, which is that they really can't do what they need to do on their own. They have to be inside a living cell. Does anybody else have any? Complex molecules. Oh wow, I may have pitched this a little too layperson. I can see we have we got some scientists in the audience. So yeah, this is kind of peak cold and flu season. I think that it's pretty salient. It's on people's minds that viruses generally make you feel sick, although that's certainly not the case of all viruses. So even though viruses are infectious particles, they don't necessarily have to cause any symptoms, or a large number of viruses don't make anything sick at all, arguably the majority. So I think a pretty good definition, if you want to pin it down, is an infectious particle which operates as an obligatory cellular, or sometimes people say molecular parasite. But one of my more favorite definitions is a piece of bad news wrapped up in a protein. And I think that kind of does summarize viruses, and we'll talk about why that is. I'm sorry, what? Obligatory. By obligatory, I mean it has to be inside a cell to complete its replicative cycle. It's a great question. Yeah, we'll get there for sure. So I think it's really important to emphasize what viruses are not, because this is such a common misconception, and I think it drives microbiologists and probably physicians crazy. Viruses are not cells. So when you think about the complexity of a cell, be it an animal cell or a plant cell or a bacterial cell, do not think about viruses. Viruses are much simpler and much smaller than any cells. So if we think about the scale of biological entities, the largest viruses actually are comparable to the smallest bacteria, and I'll be talking about some large viruses that were recently discovered. But viruses are much smaller than bacterial cells, and they're much smaller than animal cells. They're also much simpler. Viruses don't even actually look very much like cells, so we need to use electron microscopes for the most part to look at viruses, and you can see that they really have interesting structures. So on the left here, this is a pretty famous virus called Ebola virus. I think some of you might have heard of it. Pretty dangerous virus family here. And here is a bacteriophage, which actually reminds me of like a lunar landing sort of module. It's actually a molecular syringe. So these bacteriophages are viruses that infect bacteria, and they actually land on the surface of the bacteria and then will inject their their genetic contents into those bacterial cells. Rabies virus is always a favorite. It's sort of shaped like a bullet, which is, relevant since nearly everyone, almost everyone, there have been like 2 cases documented. If you get rabies, you will die of rabies if you develop symptoms. So it's shaped like a bullet, it kills you. This is a really famous virus, HIV, and polio. So you can see viruses come in all these different shapes and sizes, and they don't look like cells because they're not cells. Yeah, I'll get to in a second why they're not like cells. That's a great question. So what are you looking at here? I wanted to also emphasize how many viruses there are and how it's stars. I know, right? I actually thought about this. It does look like stars. So what you're looking at here is actually a representative milliliter of seawater, okay? And this seawater has been incubated with a molecule that makes all of the DNA present glow this fluorescent green. And the larger things are bacteria in the seawater, but these smaller these smaller particles are actually viruses present in seawater. And there's an estimated 10 to the 7th viral particles in your typical 1 mil of seawater. So the point is that there are billions and billions and billions of these viruses. I think we could argue about whether there are more stars in the universe than virus particles on Earth, but they're everywhere. And we're really just beginning to scratch the surface of what is out there in terms of the viral lifestyle, or viruses in the world. So even though viruses, they come in all these different shapes and sizes, and there's this incredible diversity of them, there's 2 basic ingredients. This kind of gets back to that quote I really liked. All viruses share in common biological information in the context of their genomes. This is maybe the thing that sometimes people, when they want to say viruses are living, they say, oh, well, it's some of them have DNA, right? And they can replicate themselves, so that's one of the ways in which they're similar to a living thing. So there's biological information in the context of a genome. It can be RNA or DNA, which is very interesting. Also single-stranded DNA, single-stranded RNA. Viruses really turn upside down a lot of what is typical, or was thought to be typical of compared to cells. And then there's the protein. The piece of bad news is wrapped inside a protein. So that's the container, the capsid. And that's what kind of confers all these really interesting shapes to the different virus particles in the world. So I'd like to emphasize that the viral life cycle is actually 2 phases. There's a dynamic intracellular phase, and then there's this inert sort of just dead, just particles floating around randomly in the world phase. That's when the infectious virions have been released. So you can really think about the viral replicative cycle or viruses in general as a biological agent with these 2 very distinct phases of existence. And so nothing is happening. They can exert no sort of agency whatsoever as virions. They're just really subject to passive movement in the world and just thermodynamic interactions with their environment. And then once they are able to attach to a cell, and are internalized. They will then undergo, depending on the virus, a whole bunch of different steps, but essentially they will take over, this is the parasite part, they'll take over that cell and use that cell's machinery and the cellular metabolism in order to make more virus particles. So that kind of gets to the crux of whether or not viruses are alive, and maybe this is a bit of a cop-out answer, but it really does depend on how you define life. And so, the kind of most commonly, the way that most people are familiar with, and the largest is this, the cell theory of life. So we probably all learned this in high school. Cells are the basic unit. Everything that's living is made up of a cell. And all cells come from previous cells. These are just some human skin cells. And so if all cells arise from other cells, this provides really the structure of the phylogenetic tree of life, the way in which we relate, how all living things are related to one another through evolution. And this is a very, very simplified diagram. I'm sure it's like for evolutionary people, it's like, oh my gosh, this is really simple. But I think it does communicate this idea really well that no matter what kind of living organism you are, we're all fundamentally related to this original progenitor cell. Sometimes it's called the last universal cellular ancestor. So you'd be thinking to yourself, well, where do viruses fit on this tree, right? I mean, they're not cells and they don't really arise necessarily from anything here. So some virologists would look at this and say, that's not the tree of life, that's the tree of viral hosts, because viruses can infect every single living thing. They can infect plants, they can infect animals, they can infect protists, archaea, bacteriophages can infect bacteria. There are even viruses upon viruses. Okay, there are viruses that parasitize other viruses. So we're really good at sequencing the genomes of these viruses and being able to relate how viruses might be related to one another, but we have a really difficult time placing them in relation to all of the other living things, right? So maybe another way of thinking about how we can sort of categorize viruses in the, I wanted to say, this is very scary territory for a microbiology graduate student to be wading into, especially since this is being videotaped. I just wanted to say, this is very controversial stuff. So here's a smattering of some back and forths between some very famous virologists. So, people argue about this. You hear some reasons why we shouldn't think about viruses in relation to living entities, right? Then these people write back, no, of course viral genomes are an important way of understanding evolution and should be included in the tree of life. No, they shouldn't be included in the tree of life. And then someone else is like, there is no such thing as a tree of life. So that's science for you. But I think another maybe useful way of thinking about how to categorize viruses is when we think about sort of what is used to be called the central dogma of biology, so the typical flow of biological information, which, by the way, viruses and what we've discovered about them has completely, changed our understanding of this, but stick with the basics here. So as you're familiar, DNA is transcribed to make RNA, which is the messenger molecule that then ribosomes, the cell's machines for making protein, will use to translate into protein. And so all viruses actually make RNA through a variety of different ways, but they all require, this is the parasitize, they have to use, they're obliged to use the host cell to translate that mRNA on cellular ribosomes. And so this is sort of a fundamental thing that all viruses share. But all living things, all cells, have their own way of making protein. So recently, folks have thought about maybe dividing up the world into two categories. You've either got the ability to make protein, you have a ribosome, and that you can make your own proteins, or you encode a capsid and you borrow other people's ribosomes and you infect these cells. So this is maybe a different way of thinking about it. Another way of thinking about are viruses alive or not is maybe to just come up with a list of properties of things that characterize living, characteristics of living things. And I think that if you were to spend some time thinking about this list, you would agree, yes, viruses can reproduce, but again, only in the context of a cell. Do viruses grow and develop? Well, insofar as they're within a cell. You sort of get the idea. Do they mutate? Yes, mostly inside cells. Well, entirely inside cells, unless you're counting like UV light and things in the environment that can mutate their genetic information. They have no metabolism of their own. They cannot make ATP. They can't metabolize any carbon molecules on their own. They absolutely are require a cell. So I think it's important to emphasize that viruses really only temporarily satisfy some of these properties of a living thing. And so it might be useful to think of them as sort of on the fringes of life. You know, they're sort of living when they're inside a cell, but when they're those virus particles, they're just inert, non-living things. So where do these amazing parasites come from? Maybe this is starting to get a little speculative. So I'd like to say it's really hard for us to know where viruses come from because they don't leave fossils. Although people are starting to look for bacterial fossils and viral fossils, but it's very, very hard, and it's kind of a new field. Also, the study of virology is actually very young. So it's only we've only like really known about viruses for about 120 years. And some of the oldest lab stocks of viral stocks that scientists have propagated over time are about 80 to 90 years old. So if you want to really look at old viruses, it'd be really useful to have some ancient viruses, or as ancient as we can get. Otherwise, we're just inferring everything from what we can get from their genomes, which is, we can get a lot. So this is really cool. People are recently looking actually at ancient water, and they're reviving bacteria and finding viruses. Remember, I told you about all those viruses in the seawater. So you have to go to very extreme places like lakes of Antarctica and Siberia, and actually they're drilling up these very deep water cores and then analyzing the genetic information present in the bacteria and in the viruses that are in that ancient water. That's kind of a cool new area. Sometimes people call it paleovirology. But there are some ideas about viral origins. There are kind of three mainstream hypotheses, and I just wanted to kind of very broadly go over them. Two of them requires or sort of assume that cells existed, that last universal common ancestor existed before viruses. And then the third kind of is independent of cells. So the progressive hypothesis is also sometimes called the escape hypothesis. And I like to think of it as a viral pickpocket. So the idea is that there's some genetic element that will arise or did arise in the cell. And it at one point becomes associated with a protein, and this is going to become its capsid. And this genetic element was then, over time as this interaction developed, acquired the ability to move to another cell. And maybe in this other cell, it acquired the ability to replicate itself, right? This is a mobile genetic element. And this idea has a lot of evidence in the sense that we observe mobile genetic elements in biology all the time. Some of you might have heard of like retrotransposons or jumping genes. Bacteria do this all the time with bacterial, plasmid horizontal gene transfer. So this is definitely something that's been seen. So maybe it happened a very, very long time ago. Oh, I meant to say, actually, the last universal common ancestor, in terms of those of you that want timelines, the estimated timeline for that in terms of when the original cell may have come about is like 4 billion years ago. So this would have been a very long time ago. The regressive hypothesis is this idea of viruses as degenerate cells. So viruses are stripped down cells, stripped down to their bare components, but they originally, according to this view, started as cells. And so in this idea, like some pre-viral cell actually developed a symbiotic relationship with a larger cell, which it infected. If you know much about mitochondria and this idea that they were once free-living bacteria that we then sort of co-opted into the animal cells, this is sort of in line with that thinking. And then over time, the cell, this symbiotic relationship, lost all of its need for metabolism, all these other things. And so actually, recently, folks have discovered what have been called giant viruses. People are trying to coin the term gyruses. I think that's awful. Let's just go with giant viruses. But Mimivirus is actually, what you're looking at here, this is a huge virus. This virus is like 3 orders of magnitude larger than the smallest virus particles that exist. It's gigantic. It's got like a 1.2 megabase. It's just genome, it's huge. It's huge for viruses. In fact, it's so big, for a long time they thought they were looking at a bacteria. That's how big it is. You can see this guy with a light microscope. And this Mimivirus, it actually infects amoebas. So remember how I told you viruses can infect all types of living cells. And what's really interesting about the genome of Mimivirus, this is just a comparison of the size compared to some other well-known viruses. Here's HIV and rhinovirus, which many different serotypes of rhinovirus cause the common cold. Mimivirus actually encodes a lot of genes that look like they came from cells, and it still definitely needs a cell to replicate, but it has sort of more of the machinery that might be necessary, so some more kind of cellular type of genes. So people sometimes think of Mimivirus, and some people have been arguing that these giant viruses might be sort of a transitional or a very ancient version of this degenerate cell virus hypothesis. And then the sort of independent theory of virus evolution, or the virus first theory, is sort of this idea that viruses evolved out of the RNA world and the primordial soup, and they didn't need any cells at all, and that they've been co-evolving with us, this entire time. And so actually kind of related, and this idea has some support, of course, in the sense that RNA, some viruses encode their genomes in RNA, and no other things do that. So it's kind of a unique feature of viruses that they can encode an entirely RNA genome. So some people think that they go back to they could go back to the original RNA world. So I put aliens in my title talk. I think that got a lot of people here. But this is actually not a very not a very mainstream hypothesis, but I thought it was worth bringing up. So some people think that it's possible. And who's going to argue that it's impossible? I guess be careful what you say. But this is panspermia, is the idea that life could transit across the universe from one place to another. And so folks have been looking for microorganisms, microscopic life. And this is the idea that maybe, since viruses are so unlike anything else that we see, and they're so difficult to classify, some people think that they might be from space. They may have come here on a comet, or on meteorites. Actually, there was a guy who published a paper in the Lancet, not a paper, like a comment, around the SARS epidemic. He was actually very seriously, positing that SARS, since it was such a novel coronavirus, maybe actually arrived from space. So that generated, as you can imagine, quite a controversy. So I'd like to just end on this idea that I wish I had a ton more time to talk about viral evolution itself, and why viruses are so interesting from an evolutionary standpoint and how they evolve. But I wanted to kind of relate this to your talk, Drew, actually. So I don't know if this number is completely right, but I read that approximately 8 million years for the changes in the genome to have accrued over time for us to have transitioned from sort of simian primates to sort of modern day Homo sapiens. If we compare that to what a virus can do, so if we look at poliovirus, which has a pretty small genome and a very high mutation rate when it replicates its genome, it can actually change 2% of its genome in only 5 days of replicative cycles, which is coincidentally the amount of time it takes for poliovirus to transit from you ingesting some contaminated food or water all the way through your gastrointestinal tract. So it kind of makes you wonder what viruses could really have done with 8 million years. And most virologists would say it's very unlikely that any of the viruses that we have today would have been around in their exact forms now. That most of the viruses that we're familiar with probably emerged around like 10,000 to 30,000 years ago. So kind of tried to get at these questions. Hopefully I've been able to persuade you that viruses are very tough to classify and that the viral world is diverse. And the origins of viruses remain mysterious, but they're fun to think about, I think. And if you're interested in learning more about viruses, or if I've piqued your interest in the world of virology, there's some really, really great resources online that are very accessible to get into. There's This Week in Virology podcast, and as well as the Virology blog. This is done by Vincent Racaniello and his friends. He's at Columbia University. He's an incredible virologist. He's kind of a great science communicator in general. He's fantastic. So this is very great content. There's also a really cool Viral Zone website. It's curated by the Swiss Bioinformatics Institute. It's really cool to look at all the different shapes and types of viruses out there. And of course, you're welcome to contact me. I really love it when people tweet me microbiology questions in general. And yeah, so thanks so much. I actually very rarely get the opportunity to speak to like a general audience, and I'd like to say like thank you for paying your taxes, because I'm funded through the NSF, and I always say that to other scientists, and it just feels kind of like, oh, you know, but really like it's so important. Thank you to fund graduate students, scientists, and all the science that's going on here. And thanks to Ask a Scientist and Wonderfest for this opportunity, and I guess we'll have a couple minutes for questions. I don't want to.
All right, who's got a question?
So if viruses are bad news, is there such a thing as a good virus?
Yeah, so there are it depends, of course, on who you ask. Oh, sorry. The question is, viruses are a piece of bad news, so are there any good news viruses, right? Is there such a thing as a good virus? There's certainly viruses that have no disease and no symptoms as far as we can tell associated with them. You know, you can argue that means that we don't know what to be looking for. Since viruses are obligatory intracellular parasites, when they hijack the cell machinery, they're fundamentally changing aspects of how that cell would function in the absence of a virus. But there are certainly some very commonly found viral infections that we can find no evidence of any disease associated with them.
So
Yeah.
answers
That's a good question.
up here.
This nice man in the, yeah.
So you mentioned that in some cases, there are viruses that infect other viruses. I find that difficult to imagine. Can you explain that?
So there are viruses that can only, sometimes these things are called satellite viruses. So these are viruses that can only infect a cell when that cell is in the process of being infected with another virus. So they're sort of like supra-infecting viruses that they are even more stripped down in what their genome encodes, such that they are parasitizing the virally encoded proteins that other virus has brought into the cell for them. So there's this, quote, fleas upon smaller fleas. So I think that's definitely true when it comes to the viral world.
Okay, one last virus question.
Maybe that's
Oh, perfect. All right.
great.
Wait, what was that? David always has great questions.
Oh, yeah.
You said that viruses were your first love. Why?
This is a great question. So, like a lot of impressionable young scientific minds, I read this book called The Hot Zone. Does anybody know about The Hot Zone?
Yes.
Yes. So this is about Ebola virus. There's also a great one, Demon in the Freezer. This is Richard Preston. Wonderful, popular science, sort of very sexy, very dangerous. That's actually what got me interested in viruses in the first place, this idea that something so simple can completely take down something as complex as the human body in just a matter of a few days. The sort of severe pathogenesis side of viruses was what got me really interested in viruses. But I've turned over to the dark side, my old scientific mentors would say, because now I study bacteria, which is like a whole nother topic. But okay, great. Thank you so much. Thanks for the opportunity. Look forward to hearing what everybody else says.
All right. From the inner spaces of viruses to the outer space of star formation through radio eyes, please welcome a spaceman from UC Berkeley, soon to be Dr. Chat Hull.
Thank you. Whoa, go for it. All right. We good? Everything's under control. All right. Very close to the mic. How's that? Sound good? All right. Really spit on it. All right, good. Thank you very much. This is it's excellent to be here. Speaking of which, I think I need to ask somebody about astrovirology, because astrobiology is a real field, not necessarily always focusing on things coming on comets or asteroids, but how extremophiles could live on other planets and other moons around Saturn and Jupiter. So it's a real thing. So keep your eye out for that. So I'm here not to tell you about that, but to tell you about star formation through radio eyes. So I'm a radio astronomer. I'm over at UC Berkeley. And I'm going to tell you about my work on magnetic fields and star-forming regions using CARMA, which is a telescope in our very own home state, which is very large. It's 7 hours away, large being the home state. But you can still be in California. So I'll show you pictures of that in a second. So just to give you a basis of what we're working with here, this is the electromagnetic spectrum. Basically, it is all that is light. Over here, you have really high-energy things like X-rays and gamma rays. And then way over here, you have super low energy, long wavelength rays of light that are in the kind of radio regime. You get the infrared here, the radio here. My telescope called CARMA works right here at about 1 millimeter or 3 millimeters. By the way, this is not even to scale. This little tiny bar is all that we can see with our eyes. Visible light is a tiny little sliver of the electromagnetic spectrum. So there's a lot more going on. Now, stars, of course, emit lots of light in the visible as well, but they also emit light elsewhere, and that's what I'm going to talk to you about. A sort of general truth of star formation is that stars form inside clouds of dust and gas, like this one. However, you might ask, well, where are the stars forming? And I could tell you that you can't actually see it in this photo. So this is a Hubble image, that's an optical image of a star-forming cloud, but you're just seeing the edge. The stars are forming inside this blob right here. Now, to give you kind of some perspective on how it is that we see this, I take you to Riyadh. So this is a dust storm over Riyadh, or as I recently discovered, this is actually known as a haboob, which is the Arabic word for strong wind. Imagine that. So this is a dust storm. Say you're one of these people coming home from work at 5 o'clock, all of a sudden, oh, I'm in a dust storm, can't see anything, great. So what do? Do you open the door to your car and get out and look around? Move to California. That is one thing you could do, that's true. What would you do to gain more information than you can see with your eyes? Radar. If you have radar in your car, sure. What do you have in your car? Turn on the radio. Look at your smartphone. Figure out something that's happening via some device that is not using optical light, right? The reason for this is because here you have your little short wavelength optical light, and it gets attenuated, it gets dimmed and blocked by really small particles that are basically the same size as the wavelength of that light. Whereas here you have your haboob dust, and you have your much longer wavelength, say centimeter or even fractions of a meter wavelength light that your cell phone and your radio are using. Cool, so you can still get through the dust. Now back to the picture. This is optical. If we go just a slightly longer wavelength to the near infrared, this is, say, instead of 500 nanometers, you might be talking about a couple of microns, something like that. You basically see the same thing, but you've gotten through that outer skin. So you compare the two. So you're seeing kind of slightly denser stuff, but you still can't really see in that deep. So go to longer wavelengths. Now you get mid-infrared. This is a lower resolution telescope, so it looks more smoothed out. But now you're really beginning to see stuff that's happening up here. This is where the stars are forming. The next step is the radio. And I'm going to show you some images of what's really going on way deep inside these things a little bit later. Question? Excellent question. The question is, why don't we see any spherical-like stuff here? It's because this is way bigger than any given star. So we're looking at blobs, at clouds where a bunch of stars may be forming. This is half of a parsec, which is basically about a light year, approximately. And any spherical blob that you would see around a star would be at, say, 100th of a parsec, so several factors of 10 smaller than that. So we're looking at larger structure. But I'll show you the tiny structure in a minute. So, I told you they're made of dust and gas. Basically, what's in these clouds is a whole bunch of hydrogen, a bunch of helium. That's really what the universe is mostly made of, about 75% hydrogen, 25% helium. There's a little bit of other stuff. These molecules here, carbon monoxide, other such interesting things. They found sugar near the galactic center last year. All kinds of weird stuff out there. It's awesome. You can use these to trace various things, including bipolar outflows, which I'll show you many images of later. Importantly for my work is dust. So I'm not a dustologist, but this stuff is pretty cool looking. I can't even begin to explain what is in these things, but these are real interstellar dust particles. And for my purposes, the most important thing about these is not only do they exist, but they also have these long axes. You see, they're not totally spherical. And each of them kind of has some preferential long axis. And what I look at is I look at dust particles that are aligned by magnetic fields. So this is my sort of crappy interpretation of what's going on out in space. Yes, before we start that question.
I saw the word olivine. The olivine is a mineral.
I'll take your word for it. So the question is, olivine is a mineral. Is this the same stuff? The answer is yes. Yeah, there are very complex compounds out there in these dust particles. Yeah. There's silicates, there's iron, there's all kinds of good stuff. Yeah. So these dust particles tend to get lined up by magnetic fields. You have magnetic fields stringing through the interstellar medium, which is the stuff out of which solar systems and stars are formed. And this basically what happens is these magnetic fields manage to line dust grains up with their long axes perpendicular to the field. We can't see fields directly with telescopes, but we can see their effect on dust. So that's what I look at. I trace magnetic fields via these lined up dust grains. So now, one way you can do this is by background starlight absorption. I'm going to show you a picture of this on the next slide. Here you have your telescope. Here you have your unpolarized background star, which emits light in any given direction that kind of wiggles up and down or left and right. But you notice that once it gets here, the component that's parallel to the long axis of the dust gets absorbed, and you get through the other one. So now you're seeing polarization. You're seeing light that is polarized, that's wiggling in one direction, and that polarization is giving you information about what the magnetic field looks like in that region. Here is a real version of what I just showed you. You have stars behind this many light-year long cloud, this long, skinny spine of a cloud, and they shine through the lined-up dust in the edges of that cloud, and you can thus infer that the magnetic field direction is perpendicular all the way down, which is pretty cool. And people had assumed that what this meant was that magnetic fields were important in actually guiding how the material formed that cloud. Basically, that, as we say, they are dynamically important. They're not getting just totally twisted up in a soup. They're strong enough to withstand the motions in the interstellar medium to guide the material to make this long, skinny thing and to kind of maintain their integrity perpendicular to the thing, to the cloud. Question?
So, you're talking about magnetic fields. I thought the glue to help the universe together was gravity.
So gravity is acting here. And in fact, that sounds dangerously close to a question a collaborator asked me when we were writing our last paper. So the question is, so he said that I thought the glue that held the universe together was gravity. That is true on scales much, much, much larger than these. Now we're talking kind of cosmological scales. It's true here too. When I say dynamically important, I mean that, so gravity's still acting here. Gravity is what is pulling the material into that spine shape, right? But the magnetic fields are not so hugely overwhelmed by gravity, turbulence, and other roiling motions that they can't help form that long structure. So gravity, but magnetic fields are still at least somewhat important. So using this as the paradigm, people thought, well, many, many, many factors of 10 smaller way inside this cloud. Now we're going to start getting toward your spherical question. Magnetic fields should still be important, right? That's what we're asking. This is the question that my research goes for, is how important are magnetic fields on very small scales where you're getting really close to the actual star that's forming, as opposed to being out in the kind of wispy cloud outside of it. To answer this question, before I answer the question with my telescope, here's what it might look like, many factors of 10 down. So this little, you see a little blob right there, that is the actual protostar, this star that is accreting material onto it, stuff is falling onto it, making the star more massive. When stars form, this dotted line is the axis of rotation of the disk. This is a disk that might, say, turn into a disk that would make our solar system around this particular star, a protoplanetary disk. There's also an outflow, a bipolar outflow or a jet-like structure that takes material out so that more stuff can fall in. And most importantly for my research, these little blue lines that pinch in here and then go back out, those are the inferred magnetic field lines. The people thought things should look like this when you look at magnetic fields on really small scales, that you should pinch into an hourglass because of gravity toward the middle, and that the axis of that hourglass should be aligned with all of the other axes. So keep this picture in mind. To see whether this was the truth, we used CARMA. CARMA is the Combined Array for Research in Millimeter Wave Astronomy. It is a millimeter wave radio telescope, and I bold here. When we observe at 1 millimeter, we can use the polarization system, which is what we're going to be looking at in a second. It's kind of a Frankenstein's monster of telescopes down near Bishop. It's in southeastern California, about 3 hours north of LA. And all of these various telescopes used to be previous telescopes and were combined, hence the C, into CARMA. It is now a 23-element array. And here's CARMA in stop motion for your intermission. This amazing movie was made by the folks at the Adler Planetarium in Chicago. So what it's doing is it's looking mostly at a science target, but it keeps moving off to look at a calibrator because the atmosphere changes about every 12 minutes. Here's the plane of the Milky Way rising there. And about every 4 to 8 hours, we move to a different science target. So right as the sun starts to come up, you're going to see them swing all the way around there. That's because they looking at a totally different science target. If you want to come up and see CARMA in action, but much more slowly than that, you can drive to Bishop and then drive up 168 into the White Mountains. It's a very, very beautiful place. Some people will stop by, so feel free. So here's me a few years ago working on the polarization system. So I spent the middle 2 years out of my 6 in grad school working on the 1 millimeter dual polarization receiver system, which is housed in the cold interior of the doers. So we house these things in the 3 Kelvin, that is 3 degrees above absolute zero stage of the innermost guts of our telescopes to keep them very cold. And what we see is not on this slide. So this, I don't quite have the talk totally memorized yet. This is the moment of Zen here. So many of you all who are in science might know that it is a point of pride to come up with a truly horrific acronym for whatever it is that you're doing. In astronomy, this is all the more true because we do surveys of all types all the time. My advisor and I were unfortunately unable to quickly come up with a terrible acronym, so we sent an email to our 25 collaborators saying, please come up with something that is at least vaguely reasonable, maybe that includes pole for polarization. And finally, after another month, our friend Mark Pound from the University of Maryland came up with telescope array doing polarization. So half the work was done. We had our acronym name. Here's the collaboration. 4 out of the 5 CARMA institutions, as well as a number of others from across the US, Canada, and Europe. And now we're not looking at this. So this was background starlight absorption. We're now looking in the super, super, super dense, vaguely spherical regions around these stars via dust emission. So these grains are still lined up by the same mechanism that I told you about before, but you're no longer looking at unpolarized light shining through, rather you're looking from emission actually being shown by the dust, like that. So now along that preferential long axis there. So we're looking at this kind of thermal polarization. And here's what we see. Question in the back. Mostly nothing. Now I'm afraid because it's going to end up on YouTube. So the collaboration in this sense is many people who were broadly interested in this topic of doing a survey of polarization in star-forming cores. Many of them had worked on various aspects of what we look at in these objects here. Some had worked on the outflows, some had worked on polarization at larger scales, and many were in the collaboration. So we kind of got all of our guns together to put in for a very, very, very large amount of time on CARMA. So it's I'd say maybe a quarter of them put in work on some aspect of the large data release paper that we put out, but mostly it was kind of bouncing ideas off of people and showing that there was the support to actually put in a huge proposal. So here we have a perhaps non-representative example of one of our sources. So this is a young isolated forming star. Here you have the blue and the red shifted meaning going away from you, blue shifted meaning coming toward you, lobes of the bipolar outflow, this jet-like structure that I talked to you about. This is not dust emission. The blue and the red are imaged in carbon monoxide emission. So they're rotational transitions of CO molecules that emit light at specific frequencies. That's what we use to look at the outflows. The question, remember, was whether the outflow axis and all these other axes were lined up with the magnetic axis. Now here, you have the little line segments are the inferred magnetic field directions. And what you see here is that not only do you have that nice hourglass shape, but the axis of that hourglass is lined up with the outflow just as expected. This was the second object we ever looked at with our system, and we saw this. However, we continued the survey and found that, in fact, no other objects looked like that. And perhaps more interesting here, you have this one that has an even more glorious looking hourglass, and yet the axis of that hourglass here is not aligned with either of the two outflows coming out of this thing. So things were getting kind of deep here. Yeah, question? No, so you're talking about the outflows, the blue and the red? That is basically a purely linear structure. It's as if I had some blob here and I had one ambulance coming toward you with a higher pitch and another ambulance going away from you with a lower pitch. It's just a jet. And we're imaging both sides of it. I didn't put in any of my slides about how that works, so this might be somewhat nebulous. But it's sort of like an ambulance. We're imaging very slightly different frequencies on either side of it, so we can tell that this one's going away and that one's coming toward us. So when I flip through these next slides here, you're going to see that, sometimes you do see the hourglass, but they're not necessarily lined up with the outflows. Here, you get magnetic field direction basically perpendicular to the outflow. Here's another one where the magnetic field and the outflow are perpendicular. In this one, you have two outflows that are basically perpendicular to one another and a magnetic field axis that isn't lined up with either of them. And this is the poster child. Two of these protostars stuck together right next to each other about, I don't know, a few tenths of a light year apart, that have basically parallel outflows and basically perpendicular magnetic fields. So all things told, together what we figured out by doing the survey is that on the whole, magnetic fields and outflows are randomly aligned, which is not what people expected to see. Furthermore, if you take a subset of those, here we have random alignment. Magnetic fields in the orange, outflows in the blue. Question? Pardon me? So the question was, do we really mean random or we don't know what the arrangement is? There are a number of caveats. By random, I mean assuming that what you see is what you get. Assuming that the magnetic field orientation really does actually look like this, you need to remember, well, one needs to know, not remember, that in our data, so we have not the highest resolution data, we're probing a specific size scale, and we're seeing all the way through these blobs. So it's what we call optically thin, meaning you're not, with your telescope, you're not slamming into a wall where you can't see any further. You're actually seeing emission all the way through. So you're basically averaging out structure that is happening inside that blob. So modulo all of those statements, the average directions of the fields and the average directions of the outflows do not seem to be tightly correlated. And the next slide here, so we have our kind of random ones here. When everything is taken as a whole, it's basically random. But we have a subset where they're perpendicular, which is exactly what we didn't expect to find. It's the exact opposite of what we thought would happen. And this is interesting because it's suggesting that in these blobs, which are rotating, that the magnetic fields may actually be weak enough to be wrapped up by rotation of the blobs, of the star-forming cores. And that suggests that magnetic fields, even on scales that are very small, getting very close to where these outflows are launched, that the fields really are playing less of a role than we had thought at this kind of final scale where you're actually forming the protoplanetary disk and the bipolar outflow. Question? So the axis of rotation, when we say they are rotating, there are a number of things that are rotating. So the clouds out of which the stars form have some general rotation axis. The disks that we presume to be in there also have a rotation axis, which we assume to be parallel to that. And what we're doing is we're probing the rotation axis of everything based on the outflow direction. Because these outflows, you have a disk, like on this table, there's a disk, and outflows generally, almost always, come perpendicular off of the disks. They're launched perpendicular to the disks. So we're using, since we don't have the resolution to see all the way down inside, we're using the outflows as probes of the rotation axis. So that's the final conclusion, that magnetic fields on small scales, you can still see them, but they really don't seem to be having as big an effect as we would have thought, starting from those much larger scales. The next step, of course, there's always a next step, ALMA. Some of you may have heard of this in the news. It is the Atacama Large Millimeter Array, the next biggest and best version of CARMA. It's in the Chilean desert on the Chacnantor Plateau at about 17,000 feet elevation up near the Bolivian border. And here's an artist's rendition of ALMA in its most extended configuration with telescopes all over the place. We just put in last week a couple of proposals to look at some of the sources I just showed you with ALMA using its polarization system, which is only just now this year coming online. So the question of what's happening at even smaller scales where you actually get down to where the disks are forming, that answer is in the future. Thank you very much.
Questions?
Why is the telescope kept at 3 Kelvin?
The question is, why is the telescope kept at 3 Kelvin? Luckily, the entire telescope is not kept at 3 Kelvin because that would require a lot of energy. So the reason that the innermost stage is kept at 3 Kelvin is simply to reduce noise. To see the, to actually receive the radiation that we need to receive at the frequencies that we work at, you need a really, 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 really cold receiver to just push down thermal emission from the stuff inside your receiver. That's right. Yeah, I mean, telescopes, the comment was that telescope is emitting what you want to look at unless you cool it down. Many things, human beings, for example, emit light, in a sense, at all different frequencies, peaked at a frequency that's proportional to the temperature that we are. And the average temperature of a telescope is basically the temperature of a person. But nonetheless, you want to cool everything down so that that spectrum of light that is emitted by the stuff right next to your receiver is as low level as possible.
You were saying in the galactic center they were measuring sugar. How do you do that? How do you see what's that far away?
Well, how do we see what's that far away?
How do you tell that it's sugar?
Oh, how do you tell it's sugar too? Okay, so I didn't do that. I believe ALMA actually did that. I think those were ALMA results. So I was not privy to those observations, but the short answer is that sugar is a molecule, just like carbon monoxide or water or anything else, and molecules emit spectral lines at very specific frequencies based on their structures. And the spectral lines of sugar were seen. That's a very basic answer, but that's basically how it works. And things that were that far away, the galactic center, actually, relatively speaking, isn't that far away. Especially when you're looking in the millimeter and longer wavelengths where you can see through all of the dust that's in the plane of the Milky Way.
Could you see it in the universal center?
The universal center. But there is no universal center. Is it here? More questions.
No hands? Was that the last question? Anyone else?
All right.
Oh, here's one.
One more.
Practically speaking, who pays for ALMA and CARMA?
We would like to thank you for paying your taxes. So yes, I should have had my thank you slide at the end. CARMA is funded by a significant NSF, National Science Foundation grant that is matched in a sense by the partner universities, including Berkeley. ALMA is a worldwide collaboration of North America, Europe, and Southeast Asia, where the North American arm is partly funded by the NSF, and the other arms are funded by their own funding agencies. But it's largely international governmental collaborations. Say again? Governmental only in the sense that the money comes from a governmental budgetary line item. So scientific. All right. Thank you very much.
All right. Thank you.
Our third talk tonight has a title that I took personally when I first read it. How to Grow a Primate Brain and Why I Should. Thank you. Coming to us from UC Berkeley is soon-to-be Dr. Drew Halley.
Hi, everyone. My name is Drew. I'm a student in anthropology. I study brain evolution, and I study at UC Berkeley. So we're going to talk today about primate brain evolution. Everybody here has, hopefully, a primate brain. And I'll try and give you an idea of how you grow one and how you evolve one, which are kind of interrelated questions. So, as I said, I study brain evolution. One of the biggest problems if you study brain evolution is that brains don't fossilize very well at all. And so if you're going to try to understand how brains evolved, you don't have any extinct species to look at their brains. You just have to sort of infer from the living brains on the planet right now how things evolved. So you look at sort of variation between all the different brains you can find and try and figure out how brains evolved generally. This is a smattering of different mammalian brains. They are to scale. So you can see that there's about 100,000 like degree of difference, or sort of scale difference between different. That's just mammalian brains. It's not even including other vertebrate brains. Here you can see a human brain in the upper right corner. We have some whale brains. There's a dolphin brain. Some different other primate brains. And this is just to give you an idea of how much variation there is just within mammals. And one of the surprising things you find within brain evolution is that despite all the differences in size and all these different brains, they're actually remarkably similar. When you look at not only functional architecture, sort of like which parts of the brain do various things, they have sort of similar functional parts, but also how they develop, which is one of the things I'll talk about today. So, size isn't everything. If any of you recognize this picture of the ENIAC computer, you can sort of imagine a time travel situation where this gentleman got a nice iPhone, and in his pocket he would have more computing power than the room he's standing in. This gives you an idea of, particularly because the computer metaphor is one of the most dominant metaphors for understanding the brain in the modern era, it gives you an idea that size isn't necessarily the biggest the most important thing to think about with brain evolution. This is one of the things we think about. We tend to think, this is certainly anthropocentric, but we think that we are the smartest species on the planet. We have language, we have symbolic thinking, we have tool use certainly beyond anything else we see in nature, but we don't have the biggest brains. You can look to cetaceans and elephants for that if you'd like. So one of the questions is, how do you sort of change how the brain is wired up in order to change how it functions, in order to get some of these things we're interested in? So one of the things people look at is relative brain size, so brain-body ratio. If you have a big body, you need a big brain to run it, generally speaking. That's because a lot of the brain does sort of housekeeping functions. It's not all cognitive sort of things. A lot of it is running your muscles, sensory information, that sort of thing. So here we have 3 different animals that are roughly the same body size, a little bit of variation there, but this is all within scalar logic, so they're close enough to compare. And you can see a porpoise has about 1,700 gram brain, a lion has about 250 grams, a crocodile about 15 grams. So it gives you a sense of how much variation in brain size you can have across animals that are roughly the same size. So this is one of the things that people are interested in. Sort of the logic, I'll get into it, here we go. So Primates are what we call highly encephalized. It turns out primates don't have the largest brains. You got elephants and whales. They don't even have the largest brain-body ratio. Humans don't have the largest brain-body ratio. Turns out very, very small mammals have higher brain-body ratios because they have really small bodies. However, primates are what we call highly encephalized. In general, primate brains are larger than we should expect for their body size. What are you expecting? As you might guess, this is a statistical argument. Essentially what they do is they take a group, in our case EQ, encephalization quotient, is you take the average for all different mammals of different sizes, and you take that line, you run a regression line through it, and if you're above the line, you have more brain than you ought to have as a self-respecting mammal. If you're below the line, you have less brain than you ought to have for a good mammal. And it turns out primates have about, on average, twice as much brain as we should expect for a mammal of any given size. So here's a very, very rough overall group of different mammalian groups. Primate brains are roughly twice as large as they should be for a mammal our size. Here you can see the sort of difference. There's a lot of, this is a log-log plot. This is sort of how a lot of what's called allometry in the study of biology is done. And these are comparing lots of different mammalian groups. So, why does body size matter? As I said, a large portion of the brain is devoted to innervating the body. So you have muscles, you have viscera, all your different organs, you have sensory organs, and depending on how big your body is, those things are going to be of varying size. You're going to have more or less musculature. You need to devote more and more brain tissue if you're going to have a big body just to running it. If a particular species has more brain than it needs, if it's highly encephalized, it may have extra neurons, was the original way this hypothesis was put, for more complex behavior. Basically, you're freeing up some cognitive space to do maybe higher order thinking or something like that. So this is also, needless to say, horrendously complicated by the fact that we try to measure intelligence in other mammals or in other species besides ourselves, which is really difficult because almost all of the ways that we've figured out how to measure intelligence even within humans, which is all messed up anyway, is even harder if you try and apply it to a non-linguistic species. So, we're going to go over a couple of the ways in which primate brains are generally different from mammal brains. First off, primate brains are built a little bit differently. Here we have a capybara. These are rodents that live down in Brazil, sort of like a giant guinea pig. If you've ever seen The Princess Bride, they are rodents of unusual size. And here you have a macaque monkey. You can see here 76 grams, 87 grams, roughly equivalent brain size, and you have dramatically different number of neurons, right? By about 4 or 5 orders of magnitude. So this turns out to be true for all the primate brains we've looked at. Primate brains have higher neuronal density than other mammals, insofar as we've checked other mammals. The other problem with this kind of research is you have to look at a lot of different species if you want to make generalized statements. And unfortunately, a lot of modern neuroscience is done by comparing humans with, let's say, mice, rats, other animals we do a lot of neuroscience research on. So, primate brains, we have a couple of different things that are also different about primate brains. We have an expanded neocortex. All primates, large and small, have disproportionately large neocortex. The neocortex is sort of, if you look at a picture of a brain, like this one, the neocortex is usually what you think of as being the brain. It's the cerebrum. It's the largest portion of the cerebrum. It's a 6-layered cerebrum. There's a couple other parts. If you've ever heard of the theory that we have a reptilian brain and a mammalian brain and then a neocortical brain, it's a very terrible theory that has since been largely discredited, so don't talk about it at cocktail parties. But the neocortex is a portion of the brain that has a lot of the stuff that we think of as being higher cognitive functions. Just to slightly disabuse you from that notion, it also involves vision and other things that we don't think of as being higher cognitive functions. So it's not quite that simple of a story, but primates have a lot of neocortex. Part of the neocortex that we have is more premotor areas. You have the motor cortex, which sort of runs right down the central sulcus that runs down the side of your brain. Premotor areas are generally involved in motor planning, so we see them activating right before you do a motor action. And we think that primates have a bigger premotor cortex because we have opposable thumbs, we have a lot of manual dexterity. All primates are very good at handling things with our hands. A lot of primates are very good at swinging around in trees. Primates initially evolved to live in an arboreal niche. which we're going to come back to. So the final thing is the addition of lateral or granular prefrontal cortex. You've probably heard a lot of research about the prefrontal cortex, generally because if you disrupt or if you injure part of your prefrontal cortex, you get some very interesting kinds of cognitive deficits. You might have heard this is sort of where the soul exists, or this is where higher cognitive functioning or your personality, that sort of thing. It's actually kind of tricky for neuroscientists to talk about the prefrontal cortex because it tends to correlate to a lot of things we think of as being free will. But you're not as a self-respecting biologist, you can't actually admit to believing in that. So, now we talk about things like motor planning or impulse control or executive functions. So keep that in mind. Impulse control tends to be my favorite way of thinking about it, sort of holding off the decision to do something on an impulse. So we have a part of the prefrontal cortex that we think is unique to primates. So you want to grow a primate brain. You're going to want a brain that's too big for your body, higher density of neurons per brain mass, high neocortical proportion, expanded premotor area, and more prefrontal cortex. How do you do it? This is a developmental biology question. How do you grow a brain like this? And how is evolution this is sort of working within the paradigm, if you've ever heard of evo-devo biology, trying to understand the evolution of development. So you look at development across lots of different species and try and figure out how evolution has changed genes that affect development that build a different body. So, is it all in the genes? I'll say no. Or no, well, sort of in a way, but it's really quite complicated and we're still working it out. So this is the 10-year anniversary, an issue of Nature of the Human Genome, the Human Genome at 10. If you can see the subtitle, Growing Pains of the Genomics Age. This issue of Nature was actually sort of about the failures of the Human Genome Project. So speaking of your taxpayer dollars and the effect to which they're curing cancer or something like that, a lot of people had a lot of hopes for the Human Genome Project and that it would sort of abolish all disease from the rest of the planet. It turns out that just knowing the sequence of the genome isn't enough to actually understand how it works or how it produces pathology or even how it produces a particular phenotype, how it builds a particular body. And that job is basically what's being hammered out by developmental biology today, trying to understand not only genes, but genes don't just correlate to a particular phenotype. They actually are really, really interrelated to one another. And it's particularly complicated to understand how they produce something like a disease, a pathology, or even just a primate brain. So, brain development is what we call plastic. This is one of my favorite neuroscience experiments that ever happened. So, this is just by way of illustration to get an idea for what I mean by plastic. Animals with overlapping visual fields. If you have two eyes that overlap with one another, you can close one eye, you'll see pretty much what you see out of the other one. They have what are called ocular dominance columns. These are in the human, in the occipital cortex, in the back of your brain. They send visual information from one eye to both sides of the brain. So if you're a frog, you don't have any of this. You don't have overlapping visual fields. Your eyes face outward. They don't overlap at all. And so for a frog, if you inject a dye into one of its eyes, it'll go back. This is kind of a special technique. It'll go back and stain one side of the brain. So one eye goes to the opposite side of the brain. The other eye goes to the other side, right? Cool. This is not what we see in any binocular species. If you have overlapping visual fields, you get this really cool phenomenon where if you inject some dye, you get these stripes along the brain. And essentially what it does is that if you're looking out, you close one eye, close the other, the visual information that comes from the same point in space ends up right next to each other in the occipital cortex. This is pretty cool because you can compare the signal from both eyes, and that's what we think three-dimensional vision is. So, I'm sorry. So, this is where the experiment gets cool. They implanted an eye, an extra eye, onto a newt. And they gave the frog binocular vision artificially. And what they found was that suddenly these stripes showed up that normally shouldn't be there. So what does that mean? The first thing it means is that the stripes aren't in the genes. They didn't change the frog's genome at all. All they did was change the kind of sensory input coming into the brain, right? So you can kind of get artificial visual field overlap and you get ocular dominance within the optic tectum, which is sort of the frog equivalent to occipital cortex. So, oh, might have missed one. So basically the general idea is that without changing the genes at all, you can actually change brain structure. All you have to do is generally change the environment that the brain grows in. This is a general idea in neuroscience that if you lose a limb early on in development, your body will compensate and use the areas of the brain maybe that would have gone to that limb to do something else. So, the developmental origins of primate encephalization. So primates are highly encephalized. We have this really big brain-body ratio. One of the cool things about this is that because we have a smaller body, we have a big brain that's trying to innervate a small body. There's a lot of extra neurons, you might say, for doing other sorts of things. Now, there's a very, very brilliant graduate student at UC Berkeley who's working on this problem right now. And here you see the green line. This is primate species. The blue line is non-primate mammalian species. What this is prenatal development of brain and body size. So if you look at primate fetuses and you look at non-primate mammalian fetuses, for example, what you'll find is that every stage of gestation, primates have about twice as much brain as other mammals do. This is really unique. So cetaceans fall on this blue line, cows, mice, you name it. And every single primate species we've looked at falls on the green line. It means that primates have about twice as much brain the whole time we gestate. And it goes all the way back to this line, which is roughly the embryonic period. If you know anything about embryogenesis, the largest embryos are about that big. This makes it very, very difficult to study what's happening prior. So this has pretty important consequences for how the brain wires itself up. The entire time we gestate in the womb, there's less body for the brain to be innervating. And when you say that an area of the brain is functional or it does a particular function, usually what you're referring to is the way it's wired up. So one of the things that's interesting about this is that you can not change all that many genes. All you need to do is change the genes that say set up brain-body proportions, and you're actually going to get a lot more consequences functionally than you would necessarily expect. You don't need to have an individual gene for each different thing that's different about the primate brain. If you do, it's sort of like the butterfly effect. You just change genes that are really, really early on, and you'll actually get differences all over the place. So, small genetic changes early on can account for lots of changes in brain connectivity and function. So, why grow a primate brain? This is the evolutionary question. Why do primates have about twice as much brain as they ought to? The answer is that there's a lot of hypotheses. We don't really know. Some of these are more popular than others. The expensive tissue hypothesis says that growing a gut and growing a brain are both very metabolically expensive, and guts are holding us back from growing very big brains. So if you go to a really simple diet where you only have to have a really small gut because you're not digesting leaves or something, you can grow a really big brain. Social brain hypothesis. It turns out absolute brain size correlates to social group size in primates. So maybe you use a big brain so that you can remember lots of other conspecifics, and remember lots of other members of your species if you need to have complex social interactions. Frugivory or home range size. Larger brain primates have larger home range. This is usually because they're frugivores and they have to remember where fruit trees are. They're not just folivores where they can go around eating leaves all day wherever they find them. They have to actually remember where the fruit tree was. Longevity hypothesis, metabolic rate hypothesis. You get the idea. I love this quote. Too many factors seem to be associated with brain size in primates. So anytime you hear somebody pitch their idea, just keep in mind there's a lot of correlates to it, and we're still sort of sorting them out. Probably the answer is some combination. However, primates have a bigger brain-body ratio while they gestate. Why reduce your body size? The other way to get a high brain-body ratio, remember it's a ratio, is to reduce the size of the body. And you can become more encephalized tomorrow if you go on a crash diet, or if you cut off one of your limbs, or something else that reduces the rest of your body, right? So, primates evolved in an arboreal niche. It turns out when we look at a lot of other species, we find that body size tends to reduce as soon as a species inhabits an arboreal niche. So, this poses kind of the interesting idea. A lot of people tend to go right towards cognitive ideas, cognitive reasons for primate brain evolution. It might not have been cognitive. That's sort of our bias as scientists. It could have been something as simple as reducing body size and brain size not reducing proportionally. So here you can see perhaps some of the things that make us unique as humans might have just come from climbing trees. So what kind of a brain is a primate brain? We have larger brains than we ought to, larger neocortical volumes, more premotor area, more prefrontal area, higher density of neurons. How? We're not sure. Small changes very early on in development, probably altering brain-body proportions and neurogenesis. Why? Again, we're not sure. Maybe to remember more faces or where fruit trees are, or maybe just to climb trees. So thank you very much. I want to thank Wonderfest, UC Berkeley, and the NSF. Thank you.
Questions? Any hands? Am I missing the hands? Okay. Okay, we'll start here.
So, if the body, having the body smaller is, so like are gorillas less intelligent than other smaller apes?
So that's a great question. One of the problems, so the question was, if having a smaller body might affect cognitive things, why would, perhaps like gorillas would be less intelligent than other animals because they have a larger body? Other primates, sure. So you can actually ask this question within humans. So within the variation, some of us have larger bodies, some of us have smaller bodies. It turns out there's a really slight difference between men and women in encephalization. There's a lot of different ways you can tackle this. Relative brain size tends not to be a very useful proxy of intelligence. Human dwarves we don't think are smarter than other humans, and human giants we don't think are less intelligent than other humans. How about hobbits? You can ask Peter Jackson about that one. So generally the logic is that particularly if it's postnatal changes, so after birth changes in body growth, that's what produces most of the variation between us in this room in our encephalization, right? And the general logic is that postnatal changes aren't as important as maybe prenatal changes. And so that's one of the reasons that we think that some of these prenatal changes to brain-body allometry might actually account for some of the really cool differences, some of the really neat things that primates tend to do behaviorally that we don't think other species do quite as well. Why primates are kind of clever mammals.
Have you heard of Terence McKenna and his stoned ape?
Sure, I know of Terence McKenna.
Talk about that. And what you think of it.
I think that a lot of evolutionary theorizing tends to be very ad hoc. So one of the problems in evolutionary theorizing is what they call just so stories. So you look at the way things are today, and you can tell about 20 different stories as to how they got that way, and you pick the one you really like, and that's the evolutionary reason. A lot of what falls under evolutionary psychology often gets critiqued for this kind of methodology. You look at human behavior today, you think of, well, it could have been adaptive to do X or Y or Z, and that becomes your theory. McKenna's work, as far as I'm familiar with it, I'm not very convinced.
Not very good. Okay.
Yeah.
Thank you.
So in your picture of all the brains, you're looking at like structures of brains and so forth, and I wonder, a lot is known now about things like how memory works and a lot of these sort of molecular processes that go on in brains, and I wonder if you are able to look at differences in any of those processes across species.
Sure. So a lot of this work, a lot of brain evolution work works at the anatomical level. They don't work at the functional level. One of the reasons behind that, there's only a handful of ways you can functionally look at brains. Some of them are kind of mean and involve needles. Other ones involve MRI. So functional differences, there is research on functional differences between species, but it tends to be limited to model species and a few others. And it's hard, even humans, we don't do invasive research on humans. So we have to, let's say, take single unit recording research from certain kinds of neuroscience with mice and try and correlate it to fMRI, functional MRI research that works with humans. And they're very, very, very different methodologies. And I think, in my opinion, it's kind of hard to compare across species functionally like that. But some really, really, really interesting research has been done on that. So, yeah.
With humans, there's a lot of talk about, here I am over here, about left brain versus right brain and, I guess, deductive reasoning or whatever versus emotional types of things. are there comparable things in other species? Are there left brain, right brain kinds of dichotomies?
I wouldn't doubt it. Again, you run into the functional question where it's difficult to actually test those kinds of things. Also, some of those dichotomies tend to be a little bit dumbed down. The left brain, right brain is one of them. Generally, kind of the pop science version of that, like, oh, I'm a left brain kind of person, is a pretty gross simplification of actually what's going on at a neuroscience level. But that being said, you certainly can see lateralization of function in certain kinds of things in other species.
So we've got time for one more brain question.
So there are developmental gestational things that happen, like gestational diabetes that change size during gestation. Are people studying that to see if, because according to your theory, you should see observable changes based on that, or not really?
So differences, let's say, in body size relative to brain size and due to a kind of disease?
Well, are difference in human gestation due to whatever goes on, and does that make a difference post-birth in a measurable way?
So there's a lot of people that study, let's say, developmental disorders in brain function, especially later in life from something that happens prenatally. So it's funny, a lot of my research actually uses some of the data that gets collected on that medical side and then combines it with sort of the evolutionary anthropology question that I'm running. I would say to some degree there's like disciplinary inertia where people are working in such different fields that they don't realize that each other's results apply to the other one's field. But I'm not aware of anybody using, let's say, human developmental disorders in brain function to make evolutionary arguments or to sort of see if there's any cognitive correlates to that kind of thing. But the other problem is the degree to which, is it an encephalization thing? Are you getting an overall smaller body and smaller brain? Or are you actually getting different proportions based on those kinds of disorders? And usually with disorders, I mean, there's a lot more going on than just say, let's say, reduced body size or something like that.
Thank you, Drew. Please give a hand to soon-to-be Dr. Drew Halley. I'd like to repeat the names of the scientists that you're meeting tonight, because I think the day will come when you will look back and say, I knew so-and-so when he or she was not yet a PhD, when he or she was not yet a professor, when he or she had not yet won that Nobel Prize. There are some great researchers here. The names again are Lauren Popov, our virologist, Chat Hull, our astronomer, Drew Halley, you just heard from, our primatologist, our anthropologist, and now it is my honor to introduce our mathematician. Yeah, it's okay. Otis Chodosh is a PhD candidate at Stanford in mathematics. His specialty is geometry. He also loves physics, and I love the title of his talk. What is the best shape? The images that I had when I heard this title probably have nothing to do. Sophia Loren, Scarlett Johansson, I'm sorry, it's true. But here to talk to us at a higher level than that is soon-to-be Dr. Otis Chodosh.
Thanks, Tucker. So, thank you all for coming. And I'm sort of afraid I have some bad news, which is I don't think I can really tell you what the best shape is. In spite of Tucker's protestation. But I'm going to use this question of what's the best shape to maybe tell you something else. And the question is really what I want to talk about is what sort of things do mathematicians think about? When I tell people I'm a mathematician, oftentimes they say, oh, do you teach? Oh, do you do research? What is research in mathematics? And so I want to try to give you guys some sort of idea of what it means to be a research mathematician. Because mathematicians do teach, but a huge part of their job is really doing research and discovering new mathematics. And maybe another question that hopefully I'll convey in my excitement about these ideas is why? Why are these things interesting? Why are they, why do you do this? So my theme today, I'll start out talking about something called the isoparametric problem. And probably no one knows what the word isoparametric means, because probably thousands of years ago some Greek mathematician made it up. And So let me break it down. So iso means the same, and parametric means perimeter, which is sort of obvious after the fact. And so if you don't remember from middle school geometry, the perimeter of a shape is the length of the boundary. So let me just give you guys a picture. So isoparametric means having the same boundary length, but really the isoparametric problem asks to maximize the area. Uh-oh. Right. Oh. There we go. Let's see. Oh, there we go. Saved. It has to maximize the perimeter, maximize the area for a fixed perimeter. So the analogy that you should keep in mind is if I give you a mile of fence and say you can have as much land as you can fence off, where are you going to put your fence? So just to make it concrete, the fence will be a mile long, so that's a fixed perimeter, and the land will be the area that you're trying to enclose. And here's our picture. So maybe the first thing you might try is a long and skinny rectangle. So I think if you think about that for a second, this is a terrible choice. You're not going to get very much land. Maybe you move on, you decide to be a little more clever. So you say, okay, I'll do like an equilateral triangle. So last night I worked out for you guys what the area of the equilateral triangle is. And you can see it's about 0.5 square miles if the perimeter is 1 mile. So okay, you're probably doing better than the long skinny triangle. But it turns out that if you do a square, we're going to get 0.6 miles. And so, just ruined my punchline. So 0.5 miles for the triangle, 0.06, sorry, for the square. I have trouble with numbers sometimes. And so if you go on, you see that sort of the more sides that we add, and the more symmetric, really, that these shapes became, the more area that we enclosed in these shapes. And I don't think I really have time to explain how to make this into a rigorous mathematical statement of the more symmetric and the more sides you have, the more area you can enclose. But I'd like to just tell you maybe that the answer really is probably what you expect, is the circle. And so the circle encloses the most area over any shape, not just the few shapes that I showed you, but any shape you can imagine, as wiggly as you want, as many sides as you want. It's the circle. The best. So I'm sorry, Tucker, it's not maybe as exciting as you were hoping. But I'd like to go on and maybe give you an idea of how do mathematicians think about new problems that might come up, given maybe our limited but some level of understanding about this isoparametric problem. And so I want to go back to a problem that showed up in a Roman story called the Aeneid. And Queen Dido was traveling along and came to start the city of Carthage. And she made a deal with the locals. And the deal was, are you sure? All right. The deal was that she could have as much land as she could enclose with an ox hide. And the first part of Dido's solution was clever, but not very mathematical, which was she ripped the ox hide apart and made long strips. And she got a huge length of rope from the ox hide. But the second part is what I really want to talk about now. And Dido realized that she could use the river as part of the boundary of her land. So she didn't have to put the ox hide along the river, so she sort of got this region for free. So let's, for simplicity, say she had half a mile of ox hide. So she did a lot of ripping. But the question is, where do you put the hide in order to get the most land, the most area? And if you think about it for a minute, it's not clear how to be the most symmetric, because now we have this fixed river, and it's ruining our old intuition about the problem. But the mathematician comes along and says, okay, how do I do this? given what I already know. And so the idea is to take a given shape and reflect, imagine reflecting across the river. So now we get a shape of length 1 mile. It enclosed maybe twice the area of what we had before. And so if you think about this, this encloses less area than the circle of the same length by what we said before. And so it turns out that really the solution again is the circle. And so we've just proven, given that the original solution to the isoparametric problem was the circle, that Dido really should use the half circle as the position of the oxide. And so I sort of want to talk a little bit, really briefly, about what a modern mathematician does, because clearly these problems, I think, are maybe from the 1900s, late 1800s, and at that point, these sorts of things were really put on a properly rigorous foundation. But nowadays, people like to think about these sorts of pictures that I'm going to show you in a second, and these are called minimal surfaces. And so they're very related to the isoparametric problem. And a minimal surface is a surface where if you wiggle it a little bit, the area increases. So it's really not obvious that if you wiggle this a little bit, the area will increase. Maybe you could wiggle it that the area decreases, but you're going to have to take my word for it. And so you should imagine that these ends, I call them, the ends of the minimal surface actually go on. So this is just a picture of a part of the minimal surface. So these have been studied for maybe the last century, but it's only really recently that we're starting to see the full picture of what does a minimal surface look like and what sort of minimal surfaces are there. So maybe I'll show you this is a pretty recent picture of a minimal surface. And so this is sort of hard to see what's going on, but now somehow there's 3 ends. There's one end here, there's one end here, and then there's one on the bottom. So it's sort of it's coming out up top, it's going here and going down. And they're joined with these holes. So I think there's 6, but 5, there's 5. And just to give you guys some flavor of what sort of things people really think about with the minimal surface is these pictures are really hard to come by. So it's hard to come up with an explicit example of such a surface. And we don't even know some simple questions like, is there a relationship between the number of ends and the number of holes? So here's a surface with 3 ends and a lot of holes. But the other direction actually is a incredibly open, nobody really knows. Can you have a lot of ends and a few holes? So this is something that people care a lot about. And people really don't have any idea of how to do this sort of thing. And the large part of the problem is that we really have a hard time coming up with examples. And I want to conclude sort of with these pictures which sort of maybe bring us back down to earth. And so I've been talking about these crazy minimal surface things, but these are actually something that everyone knows about. And minimal surfaces really are just soap bubbles. So when you take a wire and you dip it in soap, so these are some very elaborate arrangements of wire, but the soap film which spans the wire turns out to be well modeled by a minimal surface. And so studying minimal surfaces has led to a variety of understanding of things, and not just soap. It models well the interfaces between fluids and related things. And yeah, so hopefully I've given you a little bit of idea what mathematicians care about.
Questions for the mathematician?
Trying to relate the 2 talks we've heard most recently, would this mean that the roundest skulls would have the largest brains?
Yeah, that's exactly right. So that's, I guess, the question 1 dimension up. So the question I was talking about was really a 2-dimensional isoparametric problem. But it turns out that in 3 dimensions, the same question and the same answer actually is true.
Next question.
Thanks. So I'm guessing that in the soap example, it's because of surface tension that it models a minimal surface well. So could you turn that on its head and use surface tension to create more minimal surfaces by simulation, for example?
So I'm not exactly an expert in simulating minimal surfaces, but I think that something like that can be done. I'm not exactly sure, but I think that people do things like that.
Yeah, so all of this is kind of visual representation, but as a mathematician, are you working with this as visual or numeric? I mean, how are you analyzing it?
So with these sorts of minimal surfaces, actually there's been a huge revolution in our understanding once we figured out how to build them on computers. And if I go back, like this surface, I think, was only really proven on a mathematical standpoint to exist and look like this once we knew that it probably existed and probably looked like that by computers. But certainly there's other domains of mathematics where there's much less much less help given by computers or given by modeling. So a lot of it is more just you're on your own.
All right, next. Are we wrapping it up? Quick talks, quick Q&As. Wait, just a question. Tucker has a question.
I do. Otis, with the almost Dr. Chodos, with the soap films, you were talking about, you call them minimal surfaces given the perimeters. But earlier with Dido's problem and the fence problem, the question was, how do you maximize the area, the surface in a sense bounded? But so I'm not seeing the connection.
Sure. So I guess the connection is that for the isoparametric problem, you could actually talk about a similar question, which is fix the area and minimize the length. And so that's actually turns out to be the same. So you can either fix the volume and minimize the parameter, or fix the parameter and maximize the area. So that's the connection between the two regions, the two topics.
Wait, I was going to wrap it up, but we've got one more question.
I was just wondering if any of this changes if you go into hyperbolic space as opposed to geometric space.
Well, so actually this is sort of dear to me at heart. And so the question was, maybe I'll try to translate it into English, so you can also ask on the surface of a sphere, for example, and there's another really interesting space, which is the opposite maybe of a sphere, where instead of curving in a positive way, you can curve in a negative way. And this is very hard to visualize. I wish I had a good picture. But there's a whole nother sort of ambient space where you can look at minimal surfaces. And it turns out that in hyperbolic space, that maybe the answer is that there are a lot more minimal surfaces. They're much easier to find in some sense. So the flat space minimal surfaces are borderline impossible to, like they're almost too hard to find, but they're there. But there's not too many of them. In hyperbolic space, there's a lot of them, I guess. So.
All right. Please join me in thanking Otis Chodos for his talk about shape. Thank you, Otis. And